Hi, everyone. Thanks for being here and sticking around. So if we have three wonderful speakers and myself today, I should say. And this session is about the complex interplay between exercise and immune responses. So we will hear some, I think, great talks about some of that. Especially now with as you've seen with some of the talks, I was really interested seeing some of the talks throughout the conference that I was able to see, looking at a lot of homeostatic function of some resident macrophages, even in the context of other models and other diseases. So that was great to see. So I think there's that growing appreciation of the immune response into an exercise. So the first talk will be myself. I'm going to go move to that podium. And okay, so my name is Ali Abdul-Sater. I'm in the Department of Kinesiology and Health Science, or School of Kinesiology and Health Science at York University. And I will be telling you about, our lab is really focused on immune responses and specifically on inflammation. But with our wonderful colleagues in the Kin department and their interest in muscle and in exercise, we began to ask the question that everybody knows in terms of exercise affecting inflammation and other immune function. But we really wanted to dig deep into the molecular mechanism of how this effect is taking place. And we wanted to see if there's, just like you have adaptation of muscle and other tissues following exercise, we wanted to see if there's an adaptation of inflammatory cells, specifically macrophages, in response to exercise. So hopefully that's what I will be sharing with you today. So I alluded to this. I knew, this is a very basic primer into immune function, and a lot of you probably are familiar with this. There's two arms of immune responses, innate and adaptive responses. Innate, this is in terms of the system, kicks in first, relatively nonspecific, involves many, many immune cells, but the most common and famous of which, monocytes and macrophages, neutrophils, in addition to many other immune cells, dendritic cells and whatnot. Their function is really important to kickstart the adaptive response, hold the fort, sometimes clearing the infection on its own, but hold the fort, and then obviously in the later stages initiate tissue repair, which is an important reason why we have immune responses. Adaptive responses kick in later, much more specific. You're probably very familiar with them. T cells and B cells, lymphocytes, right? Make antibodies and cellular responses to clear the infection, viral, and whatever it is. Our interest is really, as I mentioned, in innate immunity and in inflammation specifically. And one hallmark of inflammatory responses is production of cytokines. Again, we've seen a lot of talks looking at cytokine production, most famous for muscle people, IL-6, but there are many other, obviously, cytokines that are involved in exercise and muscle function and inflammation. So the way innate immune cells respond to the presence of a pathogen, as I said, they're fairly nonspecific, but they do have germline encoded receptors, toll-like receptors and nod-like receptors, which are intercellular, but they could be endoplasmic, in addition to many other receptors, which sense conserved components of pathogens. So they're not really looking at a particular antigen, but conserved components like lipopolysaccharide from gram-negative bacteria, could be double-stranded RNA from viruses or single-stranded RNA, and so on and so forth. And after they sense this, there's an activation of a series of really complex molecular pathways that culminate in activation of transcription factors. Now, the most important transcription factors in inflammation or in innate responses, you have NF-kappa B, which most of you will be very familiar with, some MAP kinases, and interferon regulatory factor 3, IRF3, leading to the activation of transcription and production of cytokines, in addition to kickstarting a whole bunch of other programs, right? To, as I mentioned, important for activation of adaptive responses and recruitment of additional cells. So that's the paradigm, as I mentioned. After detection of a pathogen, you get inflammation, production of these cytokines, endothelial function, all that kind of stuff, recruitment of additional pathogens, eventually, of additional immune cells. Eventually, the goal is to clear the pathogen and kickstarting tissue repair, which is a very important function of inflammatory responses. Now, calibration of these immune responses, specifically innate responses, are extremely important because if you have weak immune responses, you obviously end up with an uncontrolled infection, and that's not something we want. But so also if you have very strong immune responses, if the innate immune response goes into overdrive, now we have a problem with catastrophic cell death and tissue damage, which is the underlying cause of many, many diseases, as we will talk about. And that's something really people, I think, understand very well these days. Now, what leads to having a balanced immune response? How do you end up with this? There's a bunch of, like many other complex diseases, whether autoimmune or metabolic diseases, it's a combination of factors, right? So you have the genetic susceptibility and environmental factors. And the one we're interested, I think most of us are interested in here, physical activity, and that's what we're looking at. So as I did mention, if you have an imbalanced inflammatory response, that's going to lead to a variety of diseases that include muscular and musculoskeletal diseases, neurological diseases, cardiovascular, like other metabolic diseases, and autoimmune diseases, and then cancer, right? So really understanding how we achieve a balanced inflammatory response, I think, is key to potentially preventing these diseases. And that's why we are interested in this in the lab. And not just us. So in the past decade or so, 2 decades maybe, there's been an explosion of research if you just put exercise and inflammation. In PubMed, you get this massive number of papers now, 1,500 almost a year. People looking at that. So that's a very important, burgeoning area of research. You can also see this is really, relationship between exercise and immunity has, I think, is rooted a long time ago in that observation that the infection risk to a respiratory infection varies with exercise intensity, if you will, in a very kind of a simplified manner. Sedentary, if that's your average, if you're a sedentary individual to infection risk, that average generally drops with a moderate intensity exercise and then increases with a high intensity exercise. Now, this is very simplistic, I understand, but that's the concept where that exercise and infection and immune function has come together. And obviously, it's been picked up, in international outlets, right, about importance of exercise and inflammation, how that regulates, as I mentioned, underlying diseases like cancer. So this is a nice review by Michael Gleason back in 2011. Now it's a bit outdated, I guess, with the advance in our understanding. But still, that how does exercise exerts, I guess, its immunomodulatory function varies depending on the tissue, right? So there's effect on muscle where IL-6 plays a major role, obviously, and IL-6 can, as we all know, has some immunostimulatory or inflammatory and sometimes anti-inflammatory, depending on the setting, depending on the combination of receptors, and you have the effect on monocytes and TNF. Adipose tissue, obviously very important, with resident macrophages in the adipose being affected by exercise and producing cytokines and adipokines, and then in addition to lymphoid organs as well. But the questions we really wanted to ask, as I alluded to in the beginning of the talk, does physical exercise cause this adaptation in inflammatory cells? And that is obviously persists for a period of time. We're not really interested in that short-term transient change that occurs after exercise. That's well documented. Our interest was, does it persist and for how long? So that's the second question. How long do these changes persist for? And then digging deeper into molecular mechanisms, metabolic changes, and potentially epigenetic effects. So our model was a simple model. Being immunologists, we did not look at tissue specifically here. What we did is we exercised some mice on a treadmill, what we called a moderate exercise intensity, based on literature. I'm happy to discuss this, for 8 weeks, so quite a prolonged model. We don't think we would see adaptation in a shorter period of time. And then we harvested the bone marrow. So we flushed the bone marrow and made macrophages a process that takes 7 days in culture, right? So any kind of transient effects, any lingering effects from exercise are gone by now, and we're really seeing if there has been long-term adaptation in those macrophages in response to the exercise that had happened. And then we looked at some pro-inflammatory cytokine expression, that's gene expression, and we do see that the exercised macrophages brought from prepared from exercise mice had a significantly blunted response to LPS. That's an inflammatory stimulus. That's IL-1 and TNF. That's not maybe super surprising. That's been shown before in certain settings, but not in this particular setting in bone marrow. We also noticed anti-inflammatory cytokine like IL-10 is also reduced in response to LPS, telling us that, maybe not too surprisingly here, that there's some sort of an upstream effect since IL-10, TNF, IL-1 are all NF-kappa B mediated. So we're thinking maybe the effect is going on NF-kappa B. And that's what we did. So if we compare bone marrow macrophages that were stimulated with LPS from sedentary versus exercise, you see that degradation of I-kappa B, which is one way to measure NF-kappa B activation. So when I-kappa B that gets degraded, NF-kappa B is activated. You see that that activation is blunted in exercise bone marrow macrophages. So in summary, we think that macrophages from exercise mice exhibit some reduced NF-kappa B induced inflammation. We'll look at the other arm of, I did mention that when you have activation of these inflammatory of macrophages, you get the NF-kappa B stimulation, but you also get the interferon, type 1 interferon, IRF3 stimulation. So look at interferon beta, and we were surprised to actually see an increased activation of interferon beta in the same macrophages with the same stimulus. So then we looked at whether this is because if there's a change in interferon regulatory factor 3, IRF3, which is upstream of interferon beta, and that was the case. So exercised bone marrow macrophages seem to have a more potent response to LPS stimulation, showing that dichotomy. Initially, we struggled with this, as in, how does that work? But then if you look, interferon, type 1 interferons have these, yes, they're pro-inflammatory, obviously. They're important for clearing viral infection, but they're known as immunomodulatory, right? So interferons regulate adaptive responses. They also regulate inflammatory responses. And if you think about macrophage polarization, if we put it in that kind of simple context, interferons has been shown to tilt that polarization into the M2, more of that anti-inflammatory phase. So we looked at that. And just to give you a bit of an intro, in a very, again, simplified sense, in vitro, you could polarize macrophages into these hyper or pro-inflammatory phenotype. If you add LPS plus interferon gamma, you get an inflammatory macrophage phenotype, which rely more on glycolysis, have upregulation of glycolytic genes like PFK2, and downstream HIF1 alpha, activation of iNOS and fatty acid synthesis, relying on fatty acid synthesis. And then if you push it to the other way, more the anti-inflammatory, regulatory, the tissue repair phenotype, which is the M2, that people do it, again, in vitro by IL-4, you end up with these anti-inflammatory cytokine like IL-10 and sometimes TGF. That relies more on oxidative phosphorylation than glycolysis, and then you have upregulation of these genes like HMOX1 and arginase-1, IRG1. In reality, in vivo, macrophages never exist probably in these two extremes. They're somewhere on that spectrum, and depending obviously on the course of infection and the type of infection. Obviously, there's a homeostatic versus inflammatory condition, so that would also vary. So we looked at those M1 associated genes, if you will, and we did see that with exercise, those genes were reduced. When we look at M2 associated genes, we see that they were increased with exercise. So it seems that with exercise macrophages, we are pushing, at least when we stimulate, those were stimulated with LPS. They were not deliberately polarized to M1 or M2. There was just LPS-stimulated macrophages, which is pushing them into the inflammatory phenotype. We do see that there's more of a reduction in that pro-inflammatory phenotype, and then pushing them more into the anti-inflammatory phenotype. See quite a big induction of arginase 1. So then we think, okay, so what's the mechanism here? What's going on? So start looking at metabolic effects. Is there some sort of metabolic effect? And this is how exercise is affecting this process. And indeed, we see that when we look at, but this is, so we get those macrophages and we put them in a flow cytometer and we measure by flow cytometry those markers of either MAP kinases, phospho-S6, which downstream of mTOR and p38 MAP kinase. And we just see there is an increased activation of these phospho markers in exercised bone marrow macrophages, indicating there's some sort of enhancement of metabolic signaling in these macrophages. So that took us to the mitochondria. Well, what if exercise is also causing some sort of a change in mitochondrial function, especially what I just told you about that M1, M2, where mitochondrial, where cellular respiration is important. We said M1 relies more on glycolysis, M2 relies more on oxidative phosphorylation. So we thought, what if mitochondrial function has been changed? So again, by flow cytometry, we measure mitochondrial, if you will, oxidation status. So we can measure mitochondrial ROS or total glutathione. And we see that there's a reduction in exercise after stimulation. We see there's a reduction in mitochondrial ROS, or what we call oxidized cells, because they have high mROS and low glutathione, and an increase in the reduced mitochondria, indicating that exercise might be affecting this mitochondrial oxidation status. We also look at mitochondrial membrane potential by a JC1 dye, which allows us to measure whether mitochondrial potential is intact or not. And we do see that there's a lower percentage of depolarized macrophages, depolarized mitochondria in macrophages from exercised mice versus sedentary mice, and that's the opposite if we look at what we call healthy mitochondria. They just, they have normal mitochondrial potential, if you will. Given that there's that effect on mitochondria, so we then measured mitochondria, cellular respiration. And to do this, we did the seahorse analysis, looking at first OCR, and then what we saw is no change on basal respiration, non-mitochondrial respiration, but we saw that there's a significant increase when we polarize these cells into the M2 phenotype, which requires mitochondrial respiration. So we see when we polarize them, there's an increase in maximal respiration and in respiratory capacity in the exercise macrophages compared to sedentary. Then we look at, that's the label. So we look at essentially extracellular acidification, PER. And this is live polarization, if you will. So here we add LPS interferon gamma to push the bone marrow macrophages into the M1 phenotype. And then we follow extracellular acidification rate. As expected, that's been shown that M1 macrophages, because these are being polarized into M1, you follow them live and you see this nice increase in essentially looking at glycolysis, right? So nice increase in extracellular acidification. But macrophages coming from exercised mice actually do increase, but not to the same extent. So there's a significant reduction in that process. So now we come to the question, how long do these effects persist for? Right? So we see those effects. How long do they persist for after we stop the exercise? So to do this, what we did is we exercised the mice the same way for 8 weeks. Then one group, we timed them so that one group would have been exercised for 8 weeks and then we sacrifice, or one group have been exercised for 8 weeks and then get 2 weeks of rest. So no exercise afterwards. For 2 weeks, then we sacrifice. Wanted to see if you let the mice sit. After that, would that effect last? And the answer is no. Most of the effects we see are gone. So in terms of the effect on mitochondrial oxidation, we're losing most of those effects. You see that the improvement in reduction in percentage of oxidized mitochondria in exercised mice is largely lost once you rest those mice. Same thing goes for same thing goes for those metabolic stimuli, so phospho-ERK and phospho-S6 and p38. That increase in metabolism is lost once you rest the mice. But we did note that some of the effects on when you polarize the cells into M1 and M2, for example, when you put LPS gamma and measure that PER, that effect seems to be persistent between exercise and exercise with some rest. So now we dig deeper into that molecular mechanism. And just really quickly introducing the concept of that trained immunity. When we talk about adaptation, this has been known for quite some time now, that macrophages, or innate immune cells, can also have some form of memory. That's not the same memory that you know of in adaptive cells, where we talk about proper memory that, and recall responses that improve. This is in innate immune cells, that was discovered in, for example, in BCG vaccination, that macrophages now, that's how BCG vaccine is thought to improve innate immune responses to a non-related illness. So if you're vaccinated with BCG, you could be infected with a different pathogen, and now you have a better response to that pathogen. And that's because macrophages, upon that first stimulus, eventually remember that, and then they have a better response the second time around when they are stimulated. And that's known to involve epigenetic modifications, metabolic reprogramming, altered responses, and these are long-term effects. And we have altered responses after exercise, we have metabolic reprogramming, and we have long-term effects. So our thinking, well, what if all that is because of metabolic reprogramming? So to do this, we did an attack-seq analysis on these macrophages, non-stimulated from exercise mice or non-exercise mice, and we look at genome-wide chromatin accessibility, see what's open and what's been changed. Overall, we see a nice separation between sedentary mice and exercise mice. If we look at the chromatin accessibility, we have a lot of regions that were more accessible or less accessible. And looking at the profile analysis, as we expect, there were a lot, I don't know if you can see them, there are a lot of metabolic signaling, immune signaling, and some mitochondrial-related pathways, which was reassuring. But then we started looking into some of those things. So this analysis is not fully completed yet. We're still working on that. Everything I'm showing you, by the way, is unpublished. But so that's so we're happy to listen to your feedback on that. But we start looking into, we did not find a single location or chromatin that was completely open or completely closed. So we see an increase or a reduction. That's what we noticed. But overall, I'm running out of time, so I got to start hurrying up here. Overall, we do see an increase in cestrin 3, which many of you might be familiar of. Cestrin belongs to this family of proteins that can improve mitochondrial biogenesis and reduce ROS production. And then when we measured actually cestrin 3 gene expression, we did find that there's an increase in cestrin 3 gene expression. Similarly, this was not when we induced with LPS, cestrin 3 expression did not go up. So that anything related to LPS induction is not stimulatory like we saw with other pro-inflammatory genes. DGK gamma, which as you mostly know, converts diacylglycerol into phosphatidic acid, also involved in metabolism, insulin responses. We also see that there's an increase in chromatin accessibility that overlap. By the way, what you see here at the bottom, sorry, I'm not explaining this very well. What you see here at the bottom is ChIP-seq from available data. So you see those peaks coincide with a ChIP-seq, indicating that they are indeed where transcription factors may bind. So that opening in the chromatin is likely to lead to increase in gene expression. And you see that in the exercise mice here, the exercise bone marrow macrophages have increased accessibility compared to sedentary, and that led to increased gene expression. I'm just going to show you two more genes real quickly. We noticed MYSM1, also clear increase in gene expression. And really recently, MYSM1 was shown to, it's a D-ubiquitinase, that was shown to de-ubiquitinate important TRAF6 and TRAF3, which are important in transcription and in the activation of inflammatory pathways. So when you increase that, we think this might lead to reduction in inflammatory pathways. And that might be one way through which exercise might be causing these anti-inflammatory effects, if you will, by increasing such if you want to call them negative regulators of inflammation. So again, here we see by qPCR induction in that gene. Last one here is a gene that was reduced that I'm really interested in, DNTTIP1. It's a component, so you see a significant reduction in the chromatin accessibility. Again, this is a ChIP-seq analysis showing that this is where it binds. This was shown in 2020 in eLife. It's a component of this HDAC that would then affect the expression of a bunch of genes. So they were interested in neurogenesis, but their top hit was immune system genes. So we're definitely going to follow up on that. We think we have some sort of interesting hits to follow up on. And so hopefully I convinced you today that exercise seems to be affecting epigenetics, inflammation, metabolism, and that's how, and there's obviously all that interplay and feedback between all these pathways. Most of the work was done by Mayuri here, Mayuri Gutherson. She's a PhD student in the lab who many of you hopefully have seen her poster. And some of the work was done initially with Ardavan Jafari, a master's student in the lab. And a lot of the work was funded by NSERC. Thank you.
Ali, nice talk.
Thanks.
I have two questions, and one which I think is actually a key experiment that you will need to do. And this is, did you transplant the bone marrow into another mouse and see whether it has a better response to damage?
Well, that's a brilliant question, for sure. Yeah, I think you're right. No, we did not do that, but that...
because, I mean, at least in my hands, whenever you isolate bone marrow-derived macrophages and stimulate them with LPS, they respond like, it's not really physiological, right?
Right, so we do use
I know it's used a lot, but...
Right. Yeah, we...
So, and then in follow-up on this, or maybe a completely different question, what's the mechanism?
I know what you're saying. So how is it doing all that, right?
Yeah, so it ends up in the bone marrow, right?
Well, it's clearly...
metabolites?
It's probably, I would think, so one of the things we're interested in, itaconate, which is a metabolite that is known to potentially push into, I don't think there's any related, nobody has shown, I don't think I've seen it with exercise. That's one of the things. So like you're saying, metabolites, we're thinking itaconate maybe because that's one of the main drivers of M2 versus M1. That's potentially one thing we could think of. But I do think it's potentially a combination, metabolites, maybe obviously some sort of hormonal changes, right, with exercise, whether that from adipokines or something in the fat cells, or obviously something maybe from the muscles, IL-6, or, and in addition to obviously steroids and other hormones that are playing a turn. But tell you the truth, we haven't, I don't know if we want to go there because that's quite a complex question yet, right? But you're right. That's where it should go.
I think that's a very interesting idea that you have, and actually to riff on what Katrin was saying, I think the bone marrow transplant is a good idea. Excuse me. And I think on top of that, it would be nice to see the correlate in vivo experiment where you challenge the mice with LPS. You could either do it, and you could do it in two modes. You can do shock, and the mice should die from shock, or if they have tolerance from the pre-training, you would have less lethality. And the other is you could actually induce tolerance by a sublethal dose of LPS, then follow with a lethal dose of LPS, and so you see protection in that mode. And you can see if exercise preconditioning also gives protection, just like a sublethal dose of LPS does. So I think it'd be interesting to move in vivo and see that some of this stuff is recapitulated from what you see with BMDMs.
100%. Thanks for that. We actually have a student who's going to do exactly that, so that she's going to start in the fall. So we wanted to see these things, right? Like we wanted to be convinced that these things are happening first. But you're right. That's the next step. Is this have any effect in vivo, right? Because that's a whole different thing.
Yeah. And one comment too, if I can make. I think it's important whenever you do mitochondrial membrane potential sensitive dyes that you don't just take the steady state incorporation at face value. So a lower membrane potential for some of these sensitive dyes could mean the mitochondria are actually healthy and functioning very well, because the functioning mitochondrion keeps the membrane potential lower than if there's dysfunction, where the membrane potential will increase. For example, if there's dysfunction in complex 4, you'll have more protons that can't, go back through. So I think what we typically do is we'll challenge the cells live in the cytometer with oligomycin or FCCP and see the sensitivity of the preloaded cells to the manipulation of the mitochondria. And then you really get sort of a capacity like doing a seahorse assay.
Yeah, thank you for that, for sure. We're definitely going to have to do that.
Yeah. Thanks.
Thank you. Okay, so our next speaker. So our next speaker is Frank Mooren from University of Witten in Germany, and he will tell us about the role of circulating microRNAs in exercise physiology.
Yes, hello everybody. First of all, I would like to thank Dr. Hood and his colleagues for inviting me here and for establishing such a really nice conference, especially after 2 years of COVID lockdown. It's a really nice experiment to be back here. Yeah, my talk is about circulating microRNAs in exercise physiology and immunology, because especially in my former field in exercise immunology, it's not so much done in this work, but I would try to give you a short overview. So just as an introduction, a short history overview about microRNAs. So the first, it's a rather new area. The first microRNAs have been investigated in the nematode C. elegans in 1993, and only about 15 years ago, microRNAs have been discovered also in mammals. So they primarily function as translational repressors. So far, about 2,000 human microRNAs have been discovered, and we know that by far the most of the proteins are coded, the proteins are under microRNA control. So in 2008, first time microRNAs, circulating microRNAs have been detected and have been known as biomarkers for malignant diseases. So my talk has 3 aspects, general aspects of microRNAs, and about circulating microRNAs and exercise, and finally the relevance of circulating microRNAs. Let's get started with the first part, the general aspects. Generation and maturation of microRNAs is given here in this slide. So microRNAs, sorry, that's it, yeah. So we have 2 microRNA locations here. We have the intergenic regions with an own promoter structure, and we have microRNAs coded in introns, which are usually co-transcribed with the respective host gain. So the microRNAs are then processed inside the nucleus, and so far various processing pathways have been identified. Then they are exported into the cytosol and form a processed together with the dicer, who cares about the loop structure, which is removed, and then this, the microRNA premature complex is put together with AGO2, which gives the major microRNA complex, which is the RNA-inducing silencing complex. So, and this can then bound on the messenger RNA. It can bind to the end. It can induce a mRNA degradation. Both have the same effect. This is a repression of mRNA translation. So we know that there is some tissue specificity of microRNAs. While many RNAs are ubiquitously expressed, some microRNAs are tissue enriched. The factor is here below 20-fold. And there are other microRNAs which are tissue specific. So here the major microRNA expression is more than 2-fold of the mean expression than in other tissues. And what we know, for example, are the myomeres, which are identified primarily in muscle cells. The cardiomeres, you see here a lot of microRNAs in the heart, vasculomeres in the vessels, blood vessels, and immunomeres, which regulate different tasks of the immune cells. You see some microRNAs involved in anti-inflammatory action, here especially microRNA 21. But on the other hand, the pro-inflammatory action with microRNA 155. Some microRNAs responsible for the differentiation of immune cells, especially also the T cell subset regulation, and of course a number of microRNAs, including microRNA 21, which are responsible in the regulation of T cell apoptosis, a part which I will show you later in this talk. With regard to immune cells, we know that the different leukocyte lineages have different microRNA profiles. And we have also an influence of, oh, sorry, I always get the. We also have hints that the profiles vary with the cell differentiation. As you heard about, one example is the macrophage polarization, the switch from M1 to M2. This is also accompanied by different changes of the microRNA profile. And microRNAs affect a number of almost all immune cell functions, such as antigen presentation, T cell receptor signaling, and toll-like receptor signaling. Exercise affects also these cellular microRNA profiles, but it's quite hard to say in which way, because we don't have so much experimental data. And we see here, for example, one or two examples from the group of Dan Cooper, who studied the effects of exercise on PBMCs and neutrophils. And you see in both cell types, the exercise addresses about almost 40 microRNAs, but there's only a very small overlap. Only 6 microRNAs were affected in the same direction after the same type of exercise. What makes the microRNAs a little bit difficult is that their functionality in signaling is rather complex. We know that, for example, one signaling pathway may be affected by several mirs. So multiple microRNAs have one target. So they can compensate in some ways for each other. On the other hand, one microRNA is also able to address several targets. So it's a kind of regulation now which is a little bit more difficult to describe, and which is in some ways different from the usual cause-effect relationship we know from normal signaling pathways. What makes the microRNAs especially interesting is that they are transported. So they are working inside the cells, of course, yes, but they have an interesting function in intercellular signaling. And here you can see the different cellular release mechanisms, which we know, the exosomes, the microvesicles, the apoptotic bodies, and also the binding to lipoproteins and other proteins inside the plasma. This figure shows you what is behind. MicroRNAs can be released by passive release and also active secretion, and they can be transported to microRNAs uptaking cells, and the blood transfer is performed in several compartments, different compartments. All of them can be affected by cellular microenvironment and, of course, like different behavior and different diseases. What is about the relevance of microRNA transport pathways? The majority of circulating microRNAs are bounded to RNA binding proteins. The release of these complexes is energy dependent. AGO2, for example, the key intracellular effector protein, is also an important microRNA binding protein in plasma. The exact release mechanism is still unclear. Some circulating microRNAs are exclusively found in cell-derived vesicles, some only in protein-associated fraction. And this you can see here in a nice study by Souza and colleagues, which investigated the transfer of microRNAs via exosomes after exercise. And you see different classes, microRNAs, which change in all three compartments. So that means in plasma, in exosomes, and intracellularly in the muscle. This was studied here. Some microRNAs which change only in muscle and plasma, or in muscle and exosomes, and some microRNAs which change only in the extracellular compartment. So we have a quite different behavior between the different type of microRNAs. So final point, let me address the methodological issues associated with microRNA measurements, and which is of relevance for the studies which I show you later. So it's quite difficult to address that we have different source materials and different methods of quantification of microRNAs. And so there are low correlations so far between different measurement platforms, arrays, and quantitative PCR. And another important point is quantification and data processing. So what is, especially when you're working with microRNAs, what is the best way to analyze your data? Are you using reference genes which you can spike into your microRNA isolation or not? So there are quite different methods performed in the literature. Now to the second point, circulating microRNAs and exercise. So here in this figure you can see a synopsis of all the different microRNAs which change, the circulating microRNAs which have been observed after acute exercise. So if you see, it's not so much compared to the number of microRNAs known. To make it a little bit more clear, a number of microRNAs, which you can find here in bold, these have been found, have been found more times than the others. So, for example, the myomere 133 has been studied more often, has been found more often than others, and also here the MER 21. And the final figure now tries to address now the different groups of microRNA families, and you can see that microRNA 133 belongs to the cardio and myomeres, and the same as microRNA 1. These have been studied in a number of studies. MicroRNA 21, which is more immunomere and vascularmere, has also been investigated a lot. The target pathways of the exercise-induced immunomeres, as you can find here in orange, if you put in this into the target pathways the programs, you can find here in the KEC pathways, especially the inflammatory pathways, which are addressed by these microRNAs, or here's the TNF receptor-associated factor pathway. So we have quite, that is what well known, which immunomeres address the exercise-induced inflammatory and anti-inflammatory. So this is now an overview about circulating microRNAs in acute exercise. We have done this in marathon runners, and we focused here on a number of microRNAs which were enhanced after the marathon run, and most of them returned to basal level after 24 hours. But a number of them, like MER 1 and MER 133, they were still enhanced after 24 hours. So to compare this with other investigators. I've included here the data of other groups, and you can see that there are some similar observations, especially here for the MIR1 and MIR133A. What is about resistance exercise? So not so much studies are available about resistance exercise, but two of them we can compare. This is a study from Savada colleagues and D'Souza from 2017. They don't have the same timelines, but they have nearly a similar exercise protocol here with resistance training, bench press and leg press and knee extensions, here more focusing on the lower extremity. But it's nearly the same amount of sets and repetitions, and it's the same intensity. So, no, sorry. And there are not so much similarities instead of these two microRNAs. This is 149 and as well number 133. But the timeline is quite different. We see here in the D'Souza group an increase 4 hours after exercise, and you see in this here in the other study we have the increases a day later, so in quite different kinetics of the microRNA in both studies. What about the mode of exercise? Here a study which compared concentric versus eccentric exercise. And it's a very early study by Bernzé and his colleagues. And you can see that here, especially after 6 hours after the exercise, the myomeres increase significantly, but still after 24 hours, everything returned to basal levels. We performed a study using EMS training, which is now going more and more in attractivity, and we use it especially for our patients. So I'm working in a department now of rehabilitation sciences, and we are working with coronary artery disease patients and with orthopedic patients. And we were thinking if this might be an interesting training method. Usually, you know this just from the gym, but we try to use it also for patients. And before we applied this, we measured the different microRNAs here after EMS training. And what you can see here is that the effect of the release of microRNA 133 and 206 can be observed for more than 72 hours after the exercise. So, of course, this is related to the extreme muscle damage we are giving here to the patients. You can see here the increase of CK, of myoglobin, and also we have an increase of CRP 3 days after the exercise test. But we think that it's not only a danger signal, it's not just a signal here of muscle damage, but we assume that it's also some kind of signal which is giving, because it's prevalent now for more than 3 days. And we applied now this technique to our patients in a recent study, which I can show you here. And the question which we have the patients only here as inpatients for about 3 to 4 weeks. So EMS training is well known to be effective after 3 months, after 6 months, also in elderly people up to 12 months of testing, of training. But we tried, is it also possible to use this technique for very deconditioned patients? So, and we included only sarcopenic patients. Here you can see the sarcopenic index for males and females according to the actual guidelines. So these were very weak patients, either from orthopedic department or with cardiological diseases. And the patients were allocated to three different study arms. It's the control group, which only performed the normal exercise training, light resistance training. Then we had the whole body EMS group and the part body EMS group, which performed the same resistance training, but received also the electrical stimulation. And here you can see the results of this study. Only 4 weeks were efficient to induce a significant improvement of the 10-second chair rise test as an easy test to perform to measure muscle strength, and which is important also for daily activity. And we have also in some isometric muscle tests a significant increase after electrical stimulation. This was, it was nearly the same, either if we used whole body EMS or only the EMS training of the limbs. So both had the same effects. So these were these were now results more focusing on the acute exercise. The next question is there an evidence for microRNA signature after chronic exercise training? And if you look here and some studies about strength training and is there a mere phenotype, I can show you two different studies. This is a study from D'Souza from 2017, and here our own study, which we made, which made me an investigation about healthy controls and endurance athletes and also strength athletes. And here I show you the comparison between untrained and strength-trained athlete, and we compared these with the data of D'Souza, and what we can find here very nicely that at least three different microRNAs are upregulated in the strength-trained athlete. This is 451, this is the 138, and the MIR31A. What about endurance training and the microRNA phenotype? Here we have some more studies available, and which we compared with our own study here. And here we have about 5 microRNAs found in nearly all of the studies. It's a 21, 103, 221, 222, and 146. So at the end of this part of the talk, I just want to give you some hint to be careful about the interpretation of data on circulating microRNAs. So what we have to be aware of is that microRNA inside the circulation is an equilibrium which between the microRNAs released from cells and microRNAs uptaken by cells. So that means that circulating microRNAs do not necessarily reflect alterations of cellular microRNAs. And to prove this, just 2 studies by Ramos and D'Souza. And you can see quite different behavior between circulatory microRNAs and muscle microRNAs. So here in this endurance exercise model in mice, you can see that after endurance training here in this highly trained group, microRNA 133 is released. It's downregulated in the muscle, and you can see here the upregulation of the transcription factor. But on the other hand, here on humans using resistant exercise, you can see that the circulatory microRNA and the muscle microRNA are regulated in the same direction. So it's quite difficult to say or to say what is going intercellularly just only from circulatory data. Now the final part of my talk is about the relevance of circulating microRNA. So the biological relevance I would like to address with 2 points. This one is the question of biomarkers. I have addressed this at the beginning of the talk, and of course as an intracellular signaling. So we know a lot of circulating microRNAs and cardiac diseases. Here in this graph you can see that there are a number of microRNAs up and down regulated. Here in green you find upregulated microRNAs, and in red the downregulated microRNAs, which can be found with certain arrhythmias, with myocardial infarction, and with certain type of cardiomyopathy. So these are the different forms of cardiomyopathy, and you see some pattern of microRNAs which can be found in these different cardiac diseases. So, and that was one reason why we asked, can microRNAs also function as some marker, as some indicator of cardiac health? So, and Bergisch in his group, and as well as our own study, we can show that some microRNAs, especially the 133, can be related to the VO2 max, so the indicator of cardiac fitness in these marathon runners. In the Bergisch group found microRNA 146 as a marker of cardiorespiratory fitness, and as well the microRNA 20. We also found that circulating microRNAs can be give some hint about the cardiac dimensions. So this is one review by Sanchez Goma, who showed that, for example, microRNAs 1 and microRNA 138 can be correlated to the diameter of the left anterior left atrial diameter. And that is an important, interesting point, because we know that especially this left atrial diameter has some prognostic role for the atrial fibrillation in later life, and this is can be correlated also to regular endurance training during the former years. In our group, we found that microRNA 136 can be related to the thickness of the septum, intraventricular septum. Circulating microRNAs and athletes' immune phenotypes. This is a part which I want to address, because we were interested for a long time about the effects of exercise on apoptosis. And what we recognized was that we have a reduced exercise-induced apoptosis in trained humans. These are data of very old data published still in 2004 by our group, and you can here see the differences of apoptotic cells after a marathon run. And this is quite different if you have a well-trained group or a less well-trained group. So in the well-trained group, we have nearly no apoptosis immediately after the marathon run and 24 hours later. And this is something which we could repeat it also in mice experiments, in animal experiments. You can see the effect of untrained, the effect of exercise in untrained and trained mice, and you see here the annexin 5 positive cells in blood and in bone marrow, and we see only the effect in the untrained animals. Circulating microRNAs and athletes' immune phenotype, we ask, is this something which is programmed in these PBMCs? And we found we could show this also in a comparison between endurance trained and untrained athletes, that the apoptosis induced by PHA was reduced in the endurance-trained athletes. Interestingly, we could not demonstrate this effect using other agents like dexamethasone or the H2O2. And when we try to compare, to correlate this with the mRNA expression of certain anti-apoptotic and pro-apoptotic factors, we could find that these factors, especially XIAP, was significantly upregulated in lymphocytes under control conditions. But the figure was quite difficult after stimulation with PHA, because in this case we had an increase of both the pro-apoptotic and the anti-apoptotic factors. And this we could find also on the microRNA level. So here you can see that during the microRNA, the circulating microRNA M21 is downregulated inside the cells, but on the other hand, here you see the circulating microRNA profile, and here we can see an upregulation of 21 and 24, which are both have an anti-apoptotic function. Transfer of microRNAs also by apoptotic bodies. This is something which we could address in another study as well. By looking on this exercise effects on apoptotic cells and on stem cells, we found that there is nearly the same regulation. We have an increase after acute exercise of apoptotic cells and the release of stem cells, but this was not evident in the trained animals. So we have no increase of apoptotic cells and no increase of stem cells after exercise. And we thought, perhaps, is there some link between both? So, and what we did, we injected the apoptotic bodies, injected apoptotic cells and apoptotic bodies here in the supernatant, and this was at least possible to induce the release of hematopoietic stem cells by just this method. So far, we didn't further characterize these apoptotic bodies. It's something which is awaiting still. The regenerative potential of microRNAs via extracellular vesicles is also a point which I want to address in the last study, which I want to show you. It's by Krenkel from the Charité in Berlin. And they used exosomes and microvesicles, so that means the medium and the small extracellular vesicles which they used from healthy controls and from cardiovascular patients with coronary artery syndrome. And this figure shows you that these microvesicles from healthy controls had the ability to re-endothelize in the in vitro experiment and enhance the endothelial cell proliferation. And on the other hand, they decrease endothelial cell death. So this is a part which they could show that the transfer of these vesicles would enhance vessel health. Interestingly, the microRNA load differs between exosomes and microvesicles, yes, but there was no difference between the two groups, the healthy controls and the patients. Finally, they looked what is the effect of exercise, and they performed in 4 weeks training of high intensity interval training and compared this just with a medical therapy. And here on this part of the figure, you can see that the medium-sized extracellular vesicle fraction was able to enhance the re-endothelialization again, and that the 4 weeks were enough to decrease the leukocyte-derived medium-sized extracellular vesicle fraction, which is an important support that exercise still is improving the anti-inflammatory role. And you can see this here in this figure, that the lymphocyte activation and release of vesicles is decreased after exercise, and we have not such pro-inflammatory stimulus of the endothelial cell. So finally, I come to my summary about circulation and microRNAs. We have a number of studies available, but different types and of exercise protocols, different technical approaches, which made it quite difficult to get a complete picture. Especially as many studies are observational in the nature, the cause relationship remains often unclear. But despite different approaches, we have some overlap of exercise-regulated single-meres, which could be found in a way may lead to the definition of a specific endurance and strength phenotype in the future. It may further enhance studies in order to enhance our understanding how exercise precisely modifies organ plasticity, as microRNAs represent a complete novel level of cellular signal transduction. There seem to be specific regulating mechanisms operative which supports the role of microRNAs also in intracellular signaling beside well-known pathways such as paracrine or hormonal control. And there are now first evidences that circulating microRNAs are involved in athletes' immune phenotype. Future studies should more carefully include the role of microRNA transport mechanisms, which seem to be important for tissue-specific effects of microRNAs. So at the end, let me say thank you to the different groups here, the actual group in University of Witten and Herdecke, with Dr. Schmitz, and the former Gießen group, especially with my friend Carsten Krüger, who performed a lot of these animal experiments on apoptosis, and the Hanover group of Thomas Thun, who is collaborating with us on the microRNAs. Thank you very much for your attention.
Okay. Well, I guess we're running a little bit behind on schedule since I don't see anyone with questions. We'll move on to the next talk. Thank you. So I want to introduce our next speaker, Dr. Richard Simpson from the University of Arizona.
Okay, thanks. Thank you for hanging around. I realize it's getting a bit late on in the day. I just want to thank the organizers, first of all, for inviting me to speak. I realize immunology is a slightly different flavor from what you're used to here, so we do appreciate you giving the field some attention. So what I'm going to talk about is I'm going to talk about the lymphocyte response to exercise, and mostly acute exercise. And we're talking about lymphocytes, we're specifically thinking about CD8 positive T cells, gamma delta T cells, natural killer cells. I really wanted to highlight some of the work that we've been doing to sort of harness this effective exercise and put it into some therapeutic applications. So in terms of the exercise immunology field, there's a lot of things we've learned about what exercise does to the immune system, over the last 20 years or so. But these are, three main studies that I'd like to highlight that I think have made pretty significant contributions. So in the context of, let me see if I can get this laser pointer going, which is this one here, isn't it? Oh, there we go. So in the context of cancer, we've already heard some talks that exercise can reduce tumor burden in mice. This is a B16 melanoma model, but we can see there's a lot more immune cell infiltrates in the tumor of exercise in animals, and in particular natural killer cells and CD8 positive T cells that contribute to the suppression of tumor growth. In the context of viral infections, we know that if we infect mice with a lethal dose of influenza, and then we put them to moderate exercise, then we can extend survival in animals compared to sedentary controls. But there does appear to be a dose response here, which Ali alluded to earlier, because if we give mice very prolonged exercise, then we can exacerbate the negative effects of influenza infection. And then some randomized control trials that have come from Jeff Woods' lab, looking at the effects of exerise training on influenza vaccine efficacy. We know that if we exercise older individuals who are notoriously difficult to vaccinate, we put them through aerobic exercise for 12 months, and then we give them the influenza vaccine at the end of the training period, then they're more likely to remain seroprotected throughout the entire influenza season compared to individuals who participated in a flexibility control group. So, of course, we've seen all these effects of exercise on immune-related diseases and situations, but, we still don't really fully understand what the mechanisms are at play. So, what is exercise really doing to the immune system that's bringing about these beneficial effects? And I'm not going to answer this question during my talk today, but I'm hoping I can at least underscore one of the proposed mechanisms by which we think exercise is enhancing immunity. So that brings me on to the acute exercise immunoenhancement hypothesis. And when we're talking about exercise, we're all interested in exercise training because they're chronic adaptations and they're considered more real adaptations to exercise. But in the context of the immune system, one of the schools of thought is that every single exercise bout we do is very important. And the reason it's very important is because our body literally mobilizes billions of T cells and NK cells and effector lymphocytes. It mobilizes large amounts of immunoglobulins and other antibodies. And then every time we exercise, these immune cells and immunoglobulins are being transported and recirculated around our body. So we're essentially increasing the immunosurveillance aspect of our immune system, and we're increasing the likelihood that our immune cells can come into contact with something that we might not want in our bodies, like a viral infected cell or a transformed cell. But during exercise, we do see that there is there's a pretty robust increase in catecholamines. So we'll see epinephrine and norepinephrine increase. We know that certain myokines are released during acute exercise. With regards to the immune system, interleukin-6, interleukin-7, and interleukin-15 are particularly important. But when we look at the lymphocyte compartment, during exercise, we see a very, very robust lymphocytosis. And the most exercise-responsive cells are the natural killer cells. So these are innate lymphocytes, so they're capable of recognizing and killing viral infected cells and also cancer cells, and they do it in a non-MHC restricted manner. So having them around in the immediate situation is very beneficial. We also see T cells are mobilized in pretty large numbers, and B cells are mobilized, but not to a great extent. And this occurs within minutes of acute exercise. You can see a 4- or 5-fold increase in NK cell numbers within 2 minutes of aerobic exercise. It's a very robust, very immediate response. But during exercise recovery, so as soon as we stop exercising, we actually see this transient lymphopenia. So the number of lymphocytes in the blood during the first few hours after exercise is actually lower than it was before exercise. And it's at this point the immune cells are believed to be trafficked into tissues. So we can find these cells in the lung, we can find them in the gut, we can find them in the muscle. And it's at this point they're believed to be doing their immune surveillance roles. And then this is when the myokines are very important. IL-6, for example, that's released from muscle during exercise is very important for directing certain lymphocyte populations to tumors, in cancer models, and various other areas in the body. So just to highlight how quickly this egress happens after exercise. So what we did here is we asked people to exercise for 30 minutes. We put an IV catheter in, and then we collected blood during the last minute of the 30-minute bout of exercise, and we've just normalized it to 100% here. And then we drew blood exactly 1, 2, 3, 4, 5, and 10 minutes after exercise, because we really wanted to characterize how quickly these cells would leave the blood compartment. And then you can see there is a steady decline. It pretty much occurs within the first 4 or 5 minutes. But the natural killer cells, which are the most exercise responsive, are also the cells that show the quickest egress. And if you look here, just after 4 minutes of seated rest, after 30 minutes of dynamic exercise, 40% of all those natural killer cells that were mobilized into the blood have already left the blood compartment. So I just want to, because we're talking about acute exercise, I'll just give an overview of the acute exercise protocol that we use in our lab, and then all the techniques that we're currently using. So essentially, we bring people in, VO2 max test them, and then a week or two later, we get them to perform this 20-minute bout of graded exercise. And every 5 minutes, we just increase the intensity of exercise. So the exercise for 5 minutes at 50%, 60, 70, and then 80% VO2 max. And depending on what the research question is, we might draw blood at every intensity of exercise, or at the very least just during the 80% stage. And then we usually collect a blood sample 1 hour after exercise cessation. When we collect immune cells, we do various different assays. We're using bulk and single-cell RNA sequencing, which I'm going to show a little bit of that data today. Our lab has been interested in a long time on the effects of exercise on viral-specific T cells. So we do peptide stimulation assays. We look at using interferon gamma ELISpot assays to identify and enumerate antigen-specific T cells. Of course, flow cytometry is prominent in any immunology lab. And then something else I'll show today as well is the work we're doing with deep T-cell receptor beta sequencing. So with this technique, we're really able to characterize at the clonal level what is going on with T cells and what antigens they're recognizing, whether or not the diversity of the T cell repertoire is increasing or decreasing with exercise. And then we also use xenogenetic mouse models. So these are immunocompromised mice that we can transplant with human cancers. And then we can look at the effects of exercise-mobilized immune cells and their ability to control these cancers in a whole organism. And then, of course, we do a lot of cell culture work also. So the studies that I'm going to show you over the next few slides is data we've mostly collected in the last couple of years. So it's unpublished or it's currently in press. The individuals we've had participate in them are just regular graduate students. So if you look at the physical characteristics here, probably very similar to most grad students in any university. We have tested about 53 people over the last couple of years. Cytomegalovirus serostatus is about half of them have CMV. CMV is one of those herpes viruses that has a very prominent impact on the immune system, so it's something we like to look at. And then some of the questions that we've had have been related to the novel coronavirus. So we've looked at individuals who had a natural coronavirus infection. We've also looked at individuals before and after COVID-19 vaccination, and I'll show some of that data also. So in terms of this mobilization, we know that it's highly regulated by the adrenergic system. So as we exercise, we release catecholamines, and those catecholamines have a direct impact on beta-2 adrenergic receptors expressed by various lymphocytes. So this is work that's we've been doing for many years in the lab, and it's evolving as we go along. But the data I'm showing you here was put together by my PhD student, Kyle Smith. And essentially what we did is we had individuals perform a 30-minute bout of acute exercise, but they performed 3 trials, and it was randomized crossover double-blind, and they consumed either a placebo, a beta-1 selective antagonist in the form of bisoprolol, or a non-selective beta antagonist in the form of nadolol. And they took these drugs or placebo 3 hours before exercise, and then during exercise, we just enumerated the cell types that were mobilized into the blood. And what we can see here, just looking at natural killer cells, is that when we block all adrenergic receptors, it inhibits the mobilization of NK cells relative to placebo. But when we just block the beta-1 adrenergic receptor, we actually increase the mobilization of natural killer cells. So what we think is going on here is just by blocking the beta-1 adrenergic receptor, we're increasing the available epinephrine to signal through the beta-2 receptor, and it's going to augment the mobilization. Yeah, and we can see that, so I'll back up a second. We can see that it does this across multiple exercise intensities. So even if we draw blood at every stage of our acute exercise protocol, we can see that the bisoprolol is increasing the mobilization relative to the placebo, whereas the nadolol is inhibiting the response. So Kyle wanted to move this into an animal model, so we thought, okay, well, we know we can regulate immune cell mobilization by manipulating the adrenergic system, but what happens when we give tumor-bearing mice these particular drugs and then put the mice to exercise? So here we've used an A20. It's a syngeneic bulb C tumor. It's a lymphoma. And then Kyle injected the lymphoma into the flank of these mice. And then the mice were given 3 different diets. So they had unmodified chow, which was our control, or they consumed chow that was laced with bisoprolol, which is the beta-1 antagonist, the exact same drug we used in the humans, or they consumed chow that was laced with nadolol, which is the non-selective beta blocker. The mice were randomized to voluntary wheel running or control for 6 weeks. And then what we can see at the end of the 6-week period is we get a huge effect of exercise on inhibition of tumor growth, so it's consistent across many tumor models. But the interesting thing in our experiment is that when we give the mice nadolol, so when we block the beta-2 adrenergic receptor, the protective effects of exercise were completely abrogated. Whereas when we block the beta-1 adrenergic receptor, the effects of exercise still remained. So it tells us that exercise is inhibiting tumor growth in a manner that is dependent on beta-2 signaling. And we're currently diving into this in more detail. We're using some beta-2 adrenergic receptor knockout mice and some beta-2 chimerisms as well to try and really figure out if that's affecting the immune cells or the tumor or a combination of both. So just diving into the human data in a little bit more detail, you know, we've known for a long time that certain immune cells are mobilized into the blood. You know, so these are T cells that might be more differentiated, so they have more of these, you know, cytotoxic type markers. We know that NK cells that express activating receptors tend to be mobilized in greater numbers with exercise. And we can see this here. You know, if you look at NK cells, this is just fold change from rested during exercise. You know, we get about 4 or 5-fold change in natural killer cells. You know, the more mature natural killer cells are more exercise responsive than the immature cells. If we look at T cells, then we get a stepwise preferential mobilization of more differentiated T cells. You know, naive T cells are not mobilized in great numbers, whereas CD45RA positive effector memory cells are mobilized in relatively large numbers. And then in you know, suppressor cells of the immune system, such as T regulatory cells, are actually not very exercise responsive. So overall, we get this more cytotoxic immune profile in the blood during exercise. We've been able to do some single-cell RNA sequencing on PBMCs collected at rest and during exercise. And you know, using the Azimuth Human PBMC Atlas, we're able to identify so many different immune cell clusters, and then we can look at differential gene expression on those clusters and do gene set enrichment analysis. So it's really allowed us to do a real in-depth, or take a real in-depth look at what exercise is doing to certain immune cell populations. And what we have here, this is just the gene expression data. So if it's red, then it's up with exercise, green is down. These are all the different immune cell subtypes that we found to have differential gene expression following exercise. And if you look up here at the top left, there's a lot of genes that are upregulated on CD8 positive T cells, like granzyme B, perforin, granzyme H, NKG7. These are all genes that are very, very important in tumor recognition and cytotoxicity. So that tells us that the CD8 T cells in particular that are going to be more potent anti-tumor cells. And the response is actually driven by the effector memory subset of CD8 positive T cells. Okay, so when we did our gene set enrichment analysis, you know, this is, we did go on KEGG terms, just have the GO terms here as well. But again, red is upregulated, green is downregulated. And we can see there's a lot of processes that are up on CD8 T cells in particular related to antigen binding, you know, response to interferon gamma, antigen recognition and processing, natural killer cell cytotoxicity, you know, chemotaxis, response to viruses. You know, there's a lot of things going on here in response to just 30 minutes of acute exercise, okay, at the single cell level. We've also looked at antigen presenting cells. You know, we see different things going on here also. And then when we look at the COD diagrams, you know, we can really see which genes are driving some of the changes in the pathways that we're seeing. So I've just highlighted natural killer cells here and CD8 positive T cells as 2 effector lymphocyte populations that we're interested in. And then we know that cytotoxicity pathways are enriched and NK cells mobilized with exercise. It's the same on CD8 positive T cells. So perforin, granzyme B, all these things I highlighted earlier tend to be driving these pathways. But something else that's quite interesting to us is we're also seeing enrichments for graft versus host disease, and we see this on NK cells and CD8 positive T cells. And if you don't know what graft versus host disease is, it's a purely man-made disease. So it's when you take allogeneic cell products, like in a bone marrow transplant or allogeneic cell therapy, and then you give these to a cancer patient who's not completely HLA matched to the donor, then those recipients are more likely to get graft versus host disease, which can be fatal in that situation. But that's important when I talk about some of the therapeutic applications in a moment. We've also been able to do some deep TCR beta sequencing. So no one's really looked at what exercise does to T cells at the clonal level. So we think this is the very first study to do this. So we've used the Immunoseq platform for this with adaptive biotechnologies. And what we found was actually not surprising. So when we collect PBMCs from individuals during exercise, we actually find that there's a more monoclonal T cell population in the blood compartment. And that's really because the T cells that are being mobilized are antigen-experienced memory T cells. So exercise is more likely to put in or mobilize these T cell clones that have previously encountered some type of antigen. And then when we take the amino acid sequences of the particular T cell clones that we're interested in, you know, we can upload these to public databases like VDJ database. And then what we found is that a lot of the T cells that are mobilized with exercise are responding to, latent herpes viruses like Epstein-Barr virus, for example, and cytomegalovirus, but we also see T cells that are reacting with influenza. And then, of course, we actually did this after the vaccine rollout, so we were finding a lot of T cells recognizing SARS-CoV-2 antigens as well. So the other thing we can do with the TCR sequencing is we do a differential abundance analysis. So this allows us to identify unique T cell clones that are exercise responsive. So here we just have rest versus exercise. Anything that's red means it's a T cell clone that's increased in frequency with exercise, where if it's blue, then it means it's decreased in frequency. And then unsurprisingly, when we look at rest versus exercise, we get a lot more T cell clones that have increased in frequency, but from when we look at exercise versus recovery, then we see that there's an egress of certain T cell clones. And what we actually found, so we had 8 individuals do these experiments, and we found that there were 562 T cell clones that were considered exercise responsive. In other words, they were significantly elevated or decreased in response to acute exercise. So this equates to about 70 clones per person. And then what we were also able to do is combine TCR sequencing with single-cell RNA sequencing. So we can look at these in tandem. And then when we took the amino acid sequences of these clones that were exercise responsive, and then we looked to see where they clustered on the azimuth map, then unsurprisingly, we found that about 80% of all these exercise responsive T cell clones were actually CD8 positive effector memory cells. We're actually doing more work at the moment. We're looking at differential gene expression just on those clones, so we'll be able to get a lot more detailed information from that. Okay, so I'm going to switch gears just a little bit and talk about SARS-CoV-2, something we're all very familiar with. We've all been impacted by it at some level over the last couple of years. And the reason I started looking at this is, my lab has always been interested in the effects of exercise on antiviral responses. We did that before COVID was even a thing. But my university told everyone that we have to shut down all human research testing unless, of course, we're doing research related to COVID-19. So, of course, I said, okay, I'll do research related to COVID-19. So that's what we did, just to keep the wheels rolling in the lab. And as you guys, I'm sure, are aware, sorry, go back a bit. As you guys, I'm sure, are aware, a lot of data was coming out very early on, providing links between physical activity and severe COVID-19 disease. So individuals who were physically active were less likely to be hospitalized, less likely to be on a ventilator, and more likely to experience just mild symptoms of COVID-19. And during the pandemic, because I was locked down and couldn't go anywhere and I was bored, I decided to write an article for Brain Behavior Immunity just to really hypothesize what exercise could be doing to the immune system in such a way that it's going to protect people from severe COVID disease. So again, go back to this acute exercise immunoenhancement hypothesis. We know that exercise mobilizes large numbers of T cells. It also mobilizes T cells that recognizes viruses. So we wanted to know if exercise would indeed mobilize T cells that recognize SARS-CoV-2 antigens. So what we did here, these experiments were done before the vaccine was available, so none of these individuals had received the vaccine. So we recruited individuals who had previously tested positive for SARS-CoV-2 at some point, or they cohabited with someone who tested positive, and then we looked in their blood and did indeed find that they'd previously been infected. And then what we do here is we just take peptide pools. So these are peptides that span the entire region of the spike protein, the membrane protein, and the nucleocapsid antigen. And we just simply add them to whole blood. And if you have any T cells that recognize those peptides, then those T cells will be activated. They'll release interferon gamma. We can spin it down and then just measure interferon gamma in the plasma via an ELISA. So it's a real simple assay to do. But we can see here in the non-infected individuals, they've never been exposed to the virus, so there's absolutely no T cell response. But the individuals that have been infected, we get a pretty robust T cell response when we stimulate with the peptide pools. And then we can see that there's a dose response, an exercise dose dependent increase in the number of SARS-CoV-2 T cells we have in circulation. Another technique we use is interferon gamma ELISpot. So this is a way to quantify the number of cells that are responding to the viral peptides. And then again, we confirmed that this response here is due to increases in number, which we can see going up with exercise. When we look at the neutralizing antibodies, we also see that they're increasing transiently with exercise before falling back down to baseline levels an hour later. And then just as a positive control in the assay, we stimulate with peptide pools against viruses that we think every one of our subjects will have been exposed to. And of course, Epstein-Barr virus is a very common herpes virus, and we can see that all of our individuals had a response to the EBV peptide pools. So when we looked at the different structural proteins in more detail, we can see that in these naturally infected individuals, the vast majority of the response was against the spike protein. It's the immunodominant antigen. It's perhaps not surprising. But we also see that T cells are responding with nucleocapsid antigen as well as membrane proteins. So we're getting similar dose response here. And again, this is our whole blood assay here, and below is just the ELISpot assay. We can see that the results are pretty comparable. And when we tested all these individuals, it was before the peak of the alpha variant in the United States, and it was actually before the first case of the delta variant. But you can see at the PBMCs, we cryopreserved these obviously, and then when the peptide pools became available, we looked at them again. And then we can see the T cells that are being mobilized with exercise are recognizing both alpha and delta variants, just suggesting that there's some cross-reactivity there of the exercise-mobilized SARS-CoV-2 T cells. We also did some TCR sequencing just on the SARS-CoV-2 specific T cells. And what we actually found is that exercise wasn't really changing the specificity of the SARS-CoV-2 response. So essentially, if you had a lot of T cells recognizing surface protein or membrane protein in the blood at rest, then those same cells tended to get mobilized into the blood. But with TCR beta sequencing, we're able to look at the entire T cells that react with the entire length of the SARS-CoV-2 genome, which is about 30,000 nucleotides. And we can see that ORF1ab and surface protein, nucleocapsid, are among the most dominant clones mobilized with exercise. And then, of course, the total number of SARS-CoV-2 clones is elevated during exercise. We also looked at the major histocompatibility complex. So I need to tell the muscle physiologist this is not mouse and heavy chains, it's a major histocompatibility complex. So CD8 positive T cells predominantly recognize peptides bound to MHC class I molecules, where CD4 T cells will recognize peptides bound to MHC class II. And then all of these clones that were mobilized with exercise were MHC1 restricted, again, just telling us that it's a CD8 T cell response that exercise is evoking. Okay, I'm really sorry about this slide. We had some technical issues with it, but please don't tell my PhD student, because I asked him to spend many long hours putting these figures together. But you'll just have to believe me when I tell you what the data says. We also looked at individuals who were vaccinated, so we tested them before and after vaccination. And then, of course, with the vaccination, we saw a neutralizing antibody response and a T cell response. But the exercise response is not as diverse in people that are vaccinated. And if you think about it, most of these individuals got the Pfizer vaccine. And it's really, really designed just to boost immune responses to spike proteins. So when we actually looked at the diversity of the T cell response, it pretty much just recognized spike protein. And there was actually no response against membrane or nucleocapsid antigen in the vaccinated people that had not previously been infected. If anyone wants to see this, I can show you on my laptop afterwards. Whereas the neutralizing antibody response to SARS-CoV-2 was actually similar between the vaccinated people and the infected people. Okay, so just to summarize, what do we know about exercise-mobilized lymphocytes? So as I mentioned earlier, we know that cytotoxic, so these are cells that are capable of recognizing and killing viral infected cells and cancer cells. These cells tend to be preferentially mobilized by exercise. So we see a really, really robust mobilization of natural killer cells, followed by gamma delta T cells, and then CD8 positive T cells. And it's these same cell subtypes that tend to leave the blood during exercise cessation. So as soon as you stop exercising, within about 4 or 5 minutes, these cells are already leaving the blood to hopefully perform their immunosurveillance duties. We know that the predominant mechanism is beta-2 adrenergic activation. We've seen that with numerous studies that have been done where we give humans different types of beta blockers and phosphodiesterase inhibitors prior to exercise, and we know there is a beta-2 dependent phenomenon. And then with the in-depth flow cytometry work that's been done, we know that these cells will have an increased expression of certain surface markers associated with cytotoxicity, co-stimulation, differentiation, and their ability to migrate through tissues. Okay, in addition, some of the work that we've added is we know that exercise is going to preferentially mobilize monoclonal T cells. So these are T cells that have pre-existing memory to some type of antigen, but mostly viruses that we have looked at. And then we also see an upregulation of transcriptomic programs associated with viral immune defense, anti-tumor immunity, antigen binding, adhesion, and their ability to respond to cytokines. And as I said, the working hypothesis is that this frequent mobilization and redistribution of these types of cells is going to increase immune surveillance in the host. But just before I finish up, I just want to talk about some of the potential therapeutic applications for this. We can mobilize and recirculate these cells in vivo, but would they actually be useful therapeutically if we were to, for example, exercise an individual, collect these cells that were mobilized into the blood, and because they have an increased ability to kill cancer cells, then transplant them to cancer patients? In this context, it could be recipients of hematopoietic stem cell transplantation that might be at a high risk of relapse. And it's actually a common procedure done. In fact, I'll just look at the slide. Here we have a patient that's received a stem cell transplant. They may have relapsed, or they might be considered high risk of relapse. So usually what happens is they'll go back to the donor, and they'll leukapheresis the donor, collect all the lymphocytes, and then adoptively transfer those lymphocytes into the patient, okay, to reduce the risk of relapse. So a very simple intervention that we're interested in is, instead of collecting these immune cells from someone at rest, is why not get them to exercise? And then we collect these exercise-mobilized cells, and then we transplant them to the patient, and hopefully that is a more efficacious response. And I don't have time to go into all these today, but what I described here are just donor lymphocyte infusions. It's probably the most rudimentary method of cell therapy. It still gets used today. But some of you may have heard of chimeric antigen receptor T cells, or CAR T cells. So these are genetically modified T cells. There's also other off-the-shelf products, gamma delta T cells we've done a lot of work with, tumor antigen-specific T cells, and cytokine-induced killer cells. But as a proof of concept for donor lymphocyte infusions, we decided to set up this experiment where we collected immune cells from people under resting conditions or during exercise. And then we used this NOD SCID mouse. So it's an NSG mouse. So it's a non-obese, non-diabetic mouse. It's got no functional immune system of its own. So it's very easy to take human cancer cells, human immune cells, transplant them to these mice, and then they will persist in this mice for up to a year, even longer. But what we did here is we use a particular type of NSG mouse. It has a human IL-15 knock-in. So this mouse will produce physiological amounts of human IL-15. And this is really important because natural killer cells, as I described earlier, they need IL-15 to survive. So if you just transplant PBMCs into a regular SCID mouse, the NK cells will be dead in like a day. But because of this knock-in mouse, it allows the NK cells to persist for many weeks. We had a number of different models here, so we use what we call the graft versus host disease model. So of course, if you take human immune cells and put them in a mouse, then those immune cells are going to recognize the mouse tissue as foreign and are going to evoke graft versus host disease. So we wanted to know what these immune cells would do in the context of GVHD without a tumor. But then we also gave the mice a luciferase tagged K562 tumor cell line. So K562 is a chronic myeloid leukemia cell line that persists in this mouse quite easily. And then, when we challenged the mice with tumor, we also adoptively transferred immune cells, and we kept the concentration of total PBMCs the same, so we gave 10 million PBMCs. But the composition of the graft does change depending on whether or not you're given resting or exercise-mobilized cells. You can see here that in the exercise-mobilized graft, we have a reduction in CD4 T cells, which are actually the major inducers of graft versus host disease. And then proportionally, we have more natural killer cells. And essentially what we found is that when these mice were, received a vehicle, then the K562 tumor grew pretty readily, after 6 weeks, whereas the adoptive transfer of the PBMCs inhibited tumor growth, but more so when the mice received PBMCs that were mobilized during exercise. And essentially what this data summarizes is that around 50% of these tumor-bearing mice that received exercise-mobilized PBMCs were alive and tumor-free after 6 weeks, compared to just 14% of mice that received resting cells. And then in our GVHD model, in the absence of tumor, there was actually no difference between resting and exercise-mobilized cells on graft versus host disease. So we think that when we're mobilizing immune cells into the blood with exercise, we think there is some clinical translational implications for this, not just for donor lymphocyte infusions, but for the ex vivo manufacture of various cell products. Okay, and with that, I'd just like to acknowledge a few individuals. I had some pictures up on the slides that was really just to underscore the individuals in my lab that were predominantly responsible for that. This is my lab at the University of Arizona. I work very closely with Manny Katsanis, who's an MD scientist, and he runs the bone marrow transplant program at the University of Arizona Cancer Center. The TCR sequencing work that you saw was predominantly done by Tiffany Zuniga. The adrenergic receptor signaling work predominantly by Kyle Smith, and then the work we did with SARS-CoV-2 was predominantly done by Forrest Baker. And then just finally, I just want to plug a meeting that's coming up in Arizona in the fall. If you are interested in exercise immunology, then the International Society of Exercise and Immunology meet every 2 years, and we will be getting together in Tucson, October 24th to 27th. Abstract submissions are open, so if you want to come and learn more about exercise immunology, then that's the meeting to be at. So thank you very much.
Well, that's just a quick question. So have you thought about your data in the context of the aging immune system, mainly because there's two well-described phenomena that happen, the increase in the memory compartment of lymphocytes, and also the switching between the ratio of CD4 to CD8 T cells. You get more CD8s the older that you are, in lymphoid tissues at least. And so that might actually have a very interesting impact whenever you look at the recruited cells after exercise in young and old people. Have you thought about that? Or?
Yeah, we have. You know, we did some studies maybe about a decade ago where we looked at immune response to acute exercise in younger and older individuals. And what we find, actually unsurprisingly, is that in older individuals, they mobilize less naive cells. Although, when you look at relative fold change, naive T cells are not that responsive to exercise, but the absolute number is still pretty large. And that's markedly reduced in older individuals, probably just virtue of the fact that they have less of them. But they do tend to mobilize more natural killer cells as a consequence of that. And then one of the confounders is the effects of latent cytomegalovirus infection. So CMV prevalence increases with age, and with CMV, you accumulate more of these exercise-responsive memory T cells. So we find that in older individuals, then their CD8 T cell response can be quite large, but it's entirely explained by CMV status.
Do you think this is a bad thing, or?
With CMV, or?
No, the overexuberant response based on the trained state of the cells in the old person with the increase in CMV and everything.
No, I don't think it is, because I think what we're what we're, at least with natural killer cells, for example, it's a very native response. And if you can still mobilize them robustly when you're older, I think that's going to be advantageous. It's still interesting why exercise tends to preferentially mobilize antigen experienced cells, and in particular cells that are specific to CMV, because the response is really quite striking.
Yeah. Well, we can talk later.
All right, yeah,
very
no
nice.
Problem, yeah.
Yeah, beautiful work, and also great to hear a Scottish accent. So, my question is, so you showed nicely that catecholamines, they stimulate the natural killer cells and have an effect on the immune system and the effect on cancer. But I mean, if you look in the literature, catecholamines essentially always seem to kill cancer patients. So in Munich, our colleagues had a paper in Cancer Cell, for example, given, and they showed that catecholamines drive pancreatic adenocarcinoma. So how do you explain that you see these beneficial effects, and all the literature is full of data showing that catecholamines kill cancer patients?
Right, it's a question I'm asked a lot, and it is a bit of a conundrum. So we published a paper in Brain Behavior and Immunity recently addressing that particular issue. And I think there's a really big difference depending on whether or not it's chronic catecholamine stimulation or whether it's just intermittent stimulation. So with exercise, it's very intermittent. But when you have chronic catecholamine stimulation, then that's when you start to see receptor desensitization, and there's a lot of other changes that go on. And then most of that data was derived from experiments done using isoproterenol. So they'll treat cells with isoproterenol in vitro for like 48 hours, 72 hours. It's a very, very long exposure. And then, of course, we see suppression of immune function. But if you actually give immune cells a quick hit, And we've done this in our lab. You just do a short-term stimulation with isoproterenol, and then you take the catecholamines away from the cells, and then you activate them. They're actually more responsive. So there's clearly a dose response or a duration of response going on.
Yeah, thank you. And also the cancer cells express adrenoreceptors and all over the place, which receptor is expressed by what cell. Yeah, beautiful work. Thank you.
Thank you. Thank you.